All right, it's my great pleasure to introduce Marina Lazic to you all today. She has started with the Master of Science in Molecular Biology from the University of Novi Sad in Serbia. At the same university, she got two Masters of Science degrees that she did at the same time, one in molecular biology and the second one in genetic plant breeding and seeds. And then she moved to the University of Wyoming, where she did a third Master of Science degree in molecular biology and also worked for a while as a research scientist. She began her PhD program at the University of Alberta in 2019, and right now is a PhD candidate in the Department of Biological Sciences at the University of Alberta in microbiology and biotechnology. So welcome, Marina. We're so happy that you're able to join us. And I'll hand it over to you.
Hi, everyone, and thanks, Carmen, for such a lovely introduction. It's my pleasure, actually, to be able to give this talk, and I hope you all will enjoy next few minutes. So my plan is actually to tell you the story about how we want to convert methane to biodegradable plastic, and I hope you will all enjoy, since I do enjoy this project. So to understand the importance of biodegradable plastic, actually at the beginning, I would first like to tell you that this biodegradable plastic that we are so much talking about is extremely environmentally friendly and recyclable material. And we are sure that if we succeed to move on with biodegradable plastic instead of traditional one, we will be able to reduce carbon emission, which is a big problem for our planet. Now, to understand the importance and significance of biodegradable plastic, we first need to understand the importance of general or traditional plastic in our lives. And I'm pretty sure that we all use plastic, or at least we used it in one point of our lives, like plastic bottles, plastic bags, not to mention medical devices and computer devices that are now actually computers parts that are made of plastic. And I guess nobody can imagine packaging without plastic material. However, our goal is to stop using this traditional plastic and to replace it with biodegradable one. Now, to understand why this matters so much, we need to understand what is traditional plastic made of. So traditional plastic that we use every day is made, or one type of plastic is made of this toxic polymer that I will shortly call PET. It has a very long name. Now, I'm sure that we all know that this PET containing traditional plastic should be discarded in these blue recycled bins. However, mistakes sometimes happen and this plastic ends up in landfill container, and the consequences are horrible. As we are piling up our waste and trash into the landfill all along with our PET containing plastic, all that trash with plastic gets exposed to the environmental factors such as rainfall. This later on creates a liquid called leachate. Now, this leachate is able to leak all to the soil and groundwater and threats to harm our wildlife. Now, I think that it's clear that we need to find solution just to avoid pollution. And solution that we are offering is replacing toxic PET with non-toxic polyhydroxybutyrate, or shortly PHB. PHB is molecule precursor for production of environmentally friendly biodegradable plastic. And my hope is that if one day we succeed to replace PET with PHB, we will end up with happy and healthy planet, which is essentially what we all want. Now, you might ask at this point, what is the problem in this solution? PHB better than PET. Let's just make plastic from PHB. The problem is that if we want to make PHB through using some microorganisms, they use some carbon source that is usually sugar-based. Where is the problem? Well, as usual, the problem is that sugar-based carbon sources are very expensive. Second problem is that if you use sugar as a carbon source to make biodegradable plastic, you are running into the food versus fuel debate because sugars are food. So we are again running into the problem, and the question is how to find solution. So solution strategy that we in my lab are offering are bacteria, microorganisms that are known as methanotrophs. What those bacteria are doing is basically acquiring a greenhouse gas methane inside of their bacterial organism, and they're transforming that methane into the PHB that is used to make biodegradable plastic. And the way how these organisms are doing is very well known. So inside of their bacterial organisms, they have a set of molecules that are called enzymes. Those enzymes are converting or transforming methane to methanol, then methanol to toxic formaldehyde, and then formaldehyde is brought up to carbon dioxide that is further released. Now, this is pretty unique mechanism among all known methanotrophs, but there is a branching point that distinguishes two main groups of these organisms, and that is the way how they assimilate this formaldehyde. One way is ramp pathway, which is a characteristic of gamma methanotrophs, and serine pathway, which is very important here, and it's done by alpha methanotrophs. And the reason why I'm pointing out this mechanism is because the organism that we are working with, Methylocystis species Rockwell, or shortly Rockwell, how I will refer to it by the end of this talk, belongs to alpha organisms. Now, these alpha organisms have serine cycle, which is a mechanism that they use to move forward with methane conversion. One of products of serine cycle is acetyl coenzyme A. This is a molecule that can be combined with another acetyl coenzyme A molecules. And then again, using molecules in organisms called enzymes, PHA, PHAB, and PHAC, they literally make PHB using this acetyl coenzyme A, and as we know, PHB is used to make biodegradable plastic. Now, I learned that whenever I'm talking about these mechanisms to the general public, people get lost. So I wanted to give a brief summary, like a mini reminder what we know so far, what's going on with these bacteria and with methane. So we have as a first main key player, greenhouse gas methane released from landfill and industry. Very bad for our planet. Second player that we are introducing now is methanol. And methanol is known as a wood alcohol. What is common for those two is that both of them are waste. So this is cheap. Nobody wants that. In addition, methane is harming us. Now we have these bacteria that I like to call superbacteria, methanotrophs, that are taking our waste. And inside of their bacterial metabolism, they are bringing them up into something that is called metabolic pathways. And you will see later on why that matters. You saw already two of those metabolic pathways. I mentioned how methane is basically brought up from methane to acetyl coenzyme A and then to the PHB. So what is happening in those metabolic pathways is basically conversions and breaking down of different molecules, starting from methane, then methane to methanol, and all the way down to PHB. And PHB is used for biodegradable plastic. Now, what we want to do to optimize this production of PHB from methane, basically, we are doing two things. First, we want to grow these organisms and to optimize biomass production. So that's the first thing. Second one is to optimize PHB production from these organisms using methane or methanol. So basically, we need to figure out how to feed them. Now, feeding can be done with carbon, nitrogen. All of these components are required for bacteria to grow. The same like for us. We need to bring carbon and nitrogen sources into our organism to grow. Now, there are other parameters that affect growth, like phosphor, pH, temperature, the same like with us. So bacteria have some optimum, and we need to find optimum to make the best growth and the best PHB production. So here is the step one. How to optimize biomass production. So we want these bacteria to grow the best possible. And first parameter that we are testing is carbon source. So here we had two possible carbon sources, methane or methanol. You remember that I mentioned a few seconds ago that these bacteria can use methane or methanol, and methane is converted to methanol inside of bacteria. Second parameter is nitrogen source. So these organisms can utilize ammonium mineral salts or nitrate mineral salts, or shorter AMS and NMS. Now, when you have these two carbon and two nitrogen sources, you can make 4 different combinations of nutrients, which are color-coded and presented here. Now, we grow bacteria under each of these 4 conditions, and we check to see how do they grow. So what this figure is showing on y-axis is time of checking, and y-axis is showing optical density, which represents a growth. So it's like indirect measurements of growth or biomass production. What I want to point out is that these organisms prefer to grow in methane, ammonium, or orange color code. So these conditions are the best for growth. Now, logical question would be, what is happening with PHB synthesis under all these 4 conditions? So that's what we did. After checking growth, we checked to see how much PHB they are making under each of these conditions. And these are results that we achieved. So what you can see on y-axis is percentage of PHB per dry weight, and on x-axis is color-coded treatment, the same like previous. And what I want to point out here is that methanol growing conditions, methanol ammonium and methanol AMS, were actually the best for PHB production. In other words, we got high growth and lower PHB, and high PHB and very low biomass. And that is something that we really don't want. What we want is to get some sweet spot between PHB and biomass. You want to find the optimum. And currently, at this moment here, we did not find the optimum because bacteria are growing the best under methane ammonium, for example, but they don't make a lot of PHB there, and vice versa. So our next question was, okay, what's happening with other molecules or metabolites in this bacterium? Because we need somehow to justify commercial production of PHB with this organism. So what we wanted to do is next. We wanted to make metabolic map of the cell. So here is what that is. So probably you all know about Google Maps. You use them every day and you're pretty much relying on them. So in the same way how Google mapped the world, we can map bacterial cell after doing something that is called global metabolomic profiling. We basically make a road map. So it's similar. we have a methane as a starting point inside of the cell, and we know what is our target destination, which is PHB production. What we don't know in this organism before we make a map is how those roads look like. Like, you remember that I mentioned those metabolic pathways, methane oxidation, how to go from methane to PHB. So we know how to go, but we don't know what's happening on those roads. Is there any obstacle? Is there traffic jam over there? So after we do this global metabolomics, we can figure that out. And these are our results. So metabolic map of the cell. Now, I know this looks very complicated, but in a few minutes, I will lead you through this and tell you what we concluded about roads inside the cell. So let's see what's happening when bacteria makes a lot of PHB, which is with methanol ammonium or yellow conditions. So under these conditions, bacteria accumulates this amino acid serine. So you see a lot of serine there, serine cycle. The reason of why is this happening is because bacteria is intoxicated apparently with methanol, and then serine is just accumulating while waiting other energetic compounds to come so the cycle can operate, the car can move down the road. Now, we don't see that acetyl coenzyme A molecule that is part of these roads, but the reason why we don't see it is because it's rapidly sent and converted to PHB. That's why we see a lot of PHB there. Now, next, low growing and high PHB conditions. Another amino acid, which is derivative of serine, and it's called glycine, is accumulated. The principle is again the same. A lot of PHB because acetyl coenzyme A is rapidly converted to PHB. Now, what's happening when bacteria grows fantastically but makes low PHB under orange growing conditions or methane ammonium? No serine, like nothing is accumulating because everything just gets degraded so fast. But we see high accumulation of amino acids, aspartate and glutamate, and that is because our acetyl coenzyme A is now shunt into another road, which leads to aspartate and glutamate. And situation is similar with green conditions, methane nitrate. We see this succinate, which is very important for later, malate or fumarate, and that is because, again, our acetyl coenzyme A is rapidly converted to those energetic metabolites. Now, what we concluded in summary, under methane growing conditions with both ammonium or nitrate, bacteria are directing their metabolism toward primary or so-called energy generating pathways. Now, under methanol growing conditions, again, with both ammonium and nitrate, bacteria are directing their metabolism towards starvation or stress-related pathways. And that's when we see PHB. PHB is like a stress-related molecule, like a supply, like when we generate supplies, when we are on restrictive caloric diet. So the question now is, how can we use this information? So here is how. It's actually very simple. There is a list of these molecules, metabolites here, that we detected with our metabolic map and global profiling. Now, some of these metabolites can be made commercially because they are very valid. They're called value-added compounds. Like, for example, amino acids. They can be used as a supplement in diet. We said PHB is used for biodegradable plastic. Succinate, which is very important, is a food additive. Aspartate and glutamate are amino acids that are often used in diets. Now, what can we do? let's say, for example, that we want to make PHB from methane or methanol using this organism. So if we want this, we would have to grow bacteria under methanol ammonium. But the bad thing here is that these conditions are actually related to the low biomass, which means starvation, and that's not paid off. You cannot justify this commercially. So if you want to industrialize PHB production using this organism, first you would have to grow bacteria under some great conditions, like a growth promoting conditions, such as methane ammonium. And then when you get enough biomass, you would stress bacteria with methanol, because methanol is starvation or stress, and then you get PHB. But here is another example. Under these high growth promoting conditions, methane ammonium, we found that there is a lot of succinate. Succinate is food additive, so it's important and it can be commercialized. This is great, because if we want to industrialize succinate or succinic acid, all what we need to do is to grow bacteria under methane ammonium, and you will get biomass and get succinate from it. What is even better if we now combine this information? You can have bioreactor where you will have our Rockwell bacterium, and you will feed Rockwell with methane, and let's say you add ammonium. That will cause Rockwell to grow very well, because Rockwell loves ammonium and methane. You can get succinate, food additive, from this. Even better, succinate is actually secreted or released from cell. So as they are growing, they just release succinate into the surrounding medium. What you can do then, use the same biomass, treat with methanol, stress cells, cause starvation, and get PHB production. Same biomass, two compounds that we can commercialize. So what is significance of all of this? I like to use simple words like killing one bird with two stones. Killing one bird, how you say that? Killing two birds with one stone. Yes, sorry, I know how this sounds in Serbian, I don't know in English. So we are solving two major environmental issues. We are solving plastic waste by replacing PET with PHB, and we are reducing methane release, which is great. Methane is harming our planet. So this picture at the end is showing my big dream, which I hope one day will happen. Maybe I won't live enough long to see, but at least I will contribute to it. So imagine to have one big landfill waste digester where we would dispose all our homemade waste. This will emit methane, and we would have one big bioreactor connected with this digester, and that bioreactor would be fulfilled with our methanotrophs that would basically acquire methane, so preventing from being emitted into the environment, and then inside of their metabolism, they will just convert it to PHB. It will be extracted and purified, and again, we can have that strategy where we would grow bacteria, have succinate excreted or any other excreted compounds, stress with methanol, and get PHB from it. So at the end, I would like to thank all of my committee members, my lab members, and my great supervisors, Lisa and Dominic, and of course my husband and son. I have to mention them. They're probably the most important for this. And at the end, I would be happy to take your questions. I hope you enjoyed.
Thank you, Marina. That was a lot of fun. Does anyone have any questions for Marina? I'm sure you have lots.
Should I stop sharing or?
You can stop, and then if you feel like you want to show a certain slide later, then go ahead.
Okay, yeah, I can go again. Okay.
So you are welcome to type in the chat. You're welcome to just turn on your microphone and ask. Vive raised a hand. So you can also do that and I will be notified. Go ahead, Vive.
Carmen. This is such an impressive and in a way, absolutely futuristic research. I thoroughly understand the importance of this work and how EFA is promoting this. But given the global impact this is going to have, how do you see this disseminated around the world so that people could readily see this research, and now that you are near completion, they would certainly see the outcome and they would want to adopt it. So this is not a technical question, but more of a marketing question in my mind. But I think it's important for you to spread the word and let people know that this is happening and you found this. I mean, you and your team. How do you see this spread around the globe?
Okay, I actually have a few ideas about that, and I recently started thinking about it, and I will tell you why. The reason why I just recently started thinking about spreading the word is because until recently, we did not have this optimized, but basically a few months ago, we found a way to optimize it. So we are in a process of publishing it. So in that moment, I believe that our plan will be to get in touch with various companies, first around Canada, there are many companies that are interested in production, especially of PHB, and not just PHB, other biopolymers. And we are currently in process of talking to them. So we are expanding it, especially because this bacterium is not the only one. There are many other methanotrophs out there that are yet to be discovered. So that's a first step. Second step that I was thinking about is basically applying for multiple conferences all around the world, including Europe, including, of course, US, Canada. I mean, I'm from Europe, so I will try to get in touch with people there, definitely. And I think one of the best ways is this type of communication that we are doing right now, especially with these online talks. I'm not marketing expert, but I believe once when we get in touch with companies that have teams who are doing that, we will be able to spread it, because we are scientists. Like, if you ask us, we find what we have, and we most of the time don't even know how to explain to general public. That's why these kind of trainings are important, and that's why we have collaborations.
Brilliant. Thank you.
We have a question in the chat, and then I think there are some people who want to ask some live. Paula asked in the chat, how do you find and isolate Rockwell?
So Rockwell is an organism that is already isolated and exists in labs that are doing methanotrophic research. So it's isolated from not from hot springs, oh my God. It's isolated, I forgot from which part of the world, but it might be somewhere where there are rice paddies in China, or basically you find methanotrophs everywhere where you see methane emissions. So let's say rice paddies would be great source, or somewhere where there are many farms because cows emit methane, or close to the landfill. So everywhere wherever you see high methane emission, you will get methanotrophs. So what we are currently working on is actually developing a rapid screening for new PHB producing methanotrophs. So that's one of my projects that I'm working on.
All right, Dan said he has a question. Dan, you want to turn on your microphone?
Yep. Hi there. So when I was in high school, I had to do a project on like converting methane into, so actually I went to school on a farm, and instead of having finals and midterms, we had projects for 10 days any individual projects or group projects. And my first project was to take like manure and to try to figure out the chemical composition to turn it into usable methane. And then for part of that project, we also went up to a dairy farm in Vermont where they had a huge biomass converter. Just they filled and they actually ran their barn electricity on the methane. So how does, I mean, I don't know if this would be something you would be able to speak to, but would that, you just said, farms, cows would be a source of methane, but is there like a convergence of, or like, which is more efficient, I guess, would be part of the question, like to convert to electricity for the methane or to convert it to biodegradable plastics, or are they something that can kind of work together?
Okay, so personally, and again, I'm not expert in electricity, so I don't know too much about that, but from what I was reading, it's way more efficient to convert methane to electricity for now. One of the problems, unfortunately, that we are running into is low efficiency. And again, that's a fact. So we don't get a lot of methane converted to PHB, but that is why we are doing that examination of metabolic pathways, because what we want to see is where is the bottleneck where our carbon goes. So let's say we see that methane is converted to acetyl coenzyme A, and acetyl coenzyme A goes to somewhere else instead of PHB. So if you know where is that bottleneck, you can improve efficiency, for example, by doing genetic engineering. You remove one gene and you overexpress the other one. But that's currently not happening. So it's way more efficient for having it for electricity. But we are continuing to work and on optimizing production from this organism and searching for new species. There are also other strategies how you can basically use methane for PHB, but I believe that's something that I shouldn't speak about because it's not published yet, and it's interesting idea. So there are all sorts of, but currently I would say that if we get good genetic engineer, we can modify organism, just cut one gene, overexpress another one, and get better methane to PHB conversion. But we are still in process of that. I don't know, does that answer your question enough? Well, but I might not be.
No, that's a good answer for sure. Thank you.
Go ahead, Roland.
Sorry, can you hear me? Because I have a difficult microphone. Okay, thanks. Yeah, thank you, Marina. That was a really interesting talk. I have a few questions and or comments because your whole technology combines many different things. And so you have, once you have the production of the plastic, by the way, I would be interested how quickly that degrades, but that's a separate thing. So you have the plastic production, but you also have the methane production. And so my concern would be that somebody says, okay, let's just do the plastic thing and get a very easily available source of methane, which is basically natural gas. And of course that would that's a fossil fuel. On the other hand, you also have, when you produce methane, methane is a worse greenhouse gas than even CO2. So if you in the process release too much methane, because you probably cannot convert it 100%, you might make the fossil, the greenhouse gas even worse. But of course, you try to solve so many problems in one go. You try to reduce the plastics, which is great, reduce the pollution that goes with the normal plastics, which is great. And then you also try to solve the greenhouse gas production. So it's a big project, and in practice, it's often it might go the wrong direction. You pointed that out with the biofuel thing that was 15 years ago a great technology until people found out that they basically produce food in order to produce. They use food in order to produce fuel, and then you cannot feed humans. So it's more of a comment, but I just wanted to point out that you might have to concentrate on one aspect at some point, and I think the bioplastic production would be the most worthwhile to start with, and then try to get the other things done too. But any comments on that?
No, sir. First of all, thank you. Great comments. And yes, I pretty much agree with everything that you just said, except I'm not concerned about us releasing methane, because I don't think that whichever process we have, which I have to point out is not happening at university, like university where I am is doing the research, and all processing technology is a step forward. So I wouldn't be concerned about releasing a methane. I don't think we can release more than it's already released because our process is degrading methane. But I agree with you that we should focus on bioplastic, and that is what we do. Just I was giving a more broad talk. Our projects are also focused, we have a few projects that are also focused on other polymers that can be made from methanol, which is also waste, and we have it a lot and it's easily available. Because whenever you have methane gas, sometimes it can be problem for gas entering to the medium because of gas mass transfer. When you have methanol, it's a liquid, it dissolves fast, it's easily available. The problems come with toxicity, but again, there are species that have very good detox strategy, and we are working with them. Some of these species are not natural PHB producers, but they can be engineered, because unlike Rockwell, they are easily engineerable. So you can make them being PHB producers. So we are focusing on that, to be honest. But great comments, thank you.
Thanks.
Thank you. There is a comment in the chat question from Jenny. Have you set up experiments to convert methane to plastics? How about converted efficiency? So maybe that's a comment about how efficiently it can happen.
Actually, yeah.
You've been speaking about a little already.
And to add to that, yes, we did. Actually, that is a novel research that I just didn't have time to talk about. That is a novel. It's still not published, hopefully will be soon. We did set up optimization experiments, and we used also statistical tools. We have statistical collaborators that are doing some modelings for us, where they basically predict where is the optimum for PHB and biomass, and then we test those experiments to see where is the best biomass and PHB, and we did that. So we are about to publish that research.
Thank you. Any more questions? And Juni says, thank you. I noticed on one of your pathways, the first methane to methanol that goes to formate and then carbon dioxide. So what happens with that carbon dioxide? Would that be released as
doesn't happen. So that's a good question, actually. But the thing with that carbon dioxide is that it often goes back into the media, and then you detect it as a carbonate ion. So it goes back to the biomass. Although Rockwell doesn't have a mechanism to fix carbon dioxide, but the amount that gets released, as far as I know, is not big, and that I know from my experiments. Very often you see only formate being excreted, so that final step of formate to carbon dioxide is not even happening. But that's not always good thing, because it's losing something called, you know that, reduction equivalents, NADHs. But still, you get formate excreted in media. What is good, that you can then engineer some heterotroph to use formate as a carbon source and make something else. So that's one of the things that we are talking about currently.
Right, because isn't formate a terminal electron acceptor?
It is, but it can be. So when formaldehyde is converted to formate, then the electron transport chain ends up there, formate gets excreted, then you end up with a lot of formate in media. And then you don't have that last step, formula to carbon dioxide, and then you have less NADHs.
Okay. Okay, thank you. There is a question in the chat from Sean Lenza. Will PHB plastics ever be able to compete with traditional plastics? What will it take to actually get to market? Does it have specific plastic applications?
Okay, so first question, I will first answer, is it going to be on the market? It is already on market. Mango Materials are making it, but not a lot. And the reason is cost. So then, which brings me to the first question, will we ever succeed to replace traditional plastic with biodegradable? I want to believe so. And I believe that if we all, I think scientists that are working in this field, if we all work on these optimizations, regardless of what strategy, doesn't matter, but there are various strategies. If we find the best way, and unfortunately we have to be able to justify like money-wise, because nobody wants to make it because traditional plastic is so cheap, so why would you bother? I think we will succeed. I can't say when, but I hope to live enough long to see that. But we shouldn't give up, is what I think. And what was the last question? Sorry.
Does it have specific plastic applications? The PHB?
Yes. Oh, actually, yes. It can be used, and it is used for medical implants, like in medicine, because it's biodegradable. So it is used now for implantations, plastic implantations. I think that some heart devices are made of PHB, polylactic acid, or hydroxypropionate, other polymers. Yeah, it needs, I just read that, yeah, needs enlightened government. Hard to get that, Roland, hard to get that.
Any more questions from the chat, or if anyone wants to open their mic, they're welcome. All right, perhaps everyone has asked their questions.
Feel free to ask. Really, I'm open for questions. I'm very passionate about this research, and I like to chat about that anytime, anywhere.
All right, I will pass it, I think, back over to Linda then to wrap up our session. Roland says thank you for a fascinating talk.
Thanks, Roland.
Thank you. Thank you, Marina. I want to, on behalf of Science Outreach, I want to thank you for doing this presentation for us. And we will have it posted on our YouTube channel, and it will be available on our website, which is scienceoutreach.ab.ca. So thank you, everyone. And Vive has said there is hope after all.
I hope so. Yes, hope never dies, at least in my case.
So thank you, everyone, and thanks for joining in. And like I said, our next presentation will be on March 24th, the Technology and Evolution of Athabasca University. Thanks again.
Thank you, guys.
Thank you.